Sequencing a complete genome produces a massive, unintelligible string of characters, but it provides no immediate functional map. To generate usable information, computational biologists must parse raw sequencer output into specific data structures. The two primary formats used in this process are FASTQ and FASTA files. A FASTQ file contains billions of raw nucleotides paired directly with the sequencing instrument's confidence scores for every single base. For the task of gene prediction, this quality data is discarded. The file is stripped down into a pure FASTA format, leaving only the sequence. A pure FASTA nucleotide string requires an algorithm capable of locating the functional genes hidden within the surrounding genomic noise. The difficulty of finding those genes depends entirely on the organism. Prokaryotic genomes are relatively simple, with highly dense coding regions. Eukaryotic genomes are sprawling and fragmented. Eukaryotic genomes operate on a massive scale. The human genome alone spans approximately 3 billion base pairs. This chart shows the composition of the human genome. The actual coding density, the exons that provide instructions for building proteins, makes up a tiny 3% slice of the total sequence, compared to the massive 97% non-coding remainder. That 97% majority consists of vast tracts of non-functional data, including massive intronic sequences, dead pseudogenes, and sprawling intergenic space. Simple rule-based computational parsers fail completely when trying to navigate this highly fragmented landscape. They cannot differentiate a functional exon from random intergenic noise. To solve this, bioinformatics relies on two competing but complementary computational paradigms: similarity searches and ab initio prediction. Gene finding in eukaryotes is a complex filtering problem. Algorithms must eliminate 97% of the genomic noise to isolate the 3% functional signal. The first major paradigm is the similarity-based search. This method operates entirely on empirical biological evidence. The core biological assumption driving this algorithm is the evolutionary conservation of functional regions. Because exons code for critical proteins, they are under strict evolutionary pressure. Any severe mutation is often lethal, meaning these sequences remain structurally stable across millions of years of evolution. Conversely, non-functional regions like introns and intergenic spaces are free to mutate rapidly without harming the organism. The algorithm takes the unannotated input sequence and systematically aligns it against massive databases of known proteins or expressed sequence tags, known as ESTs. An EST is a short subsequence of a transcribed protein-coding nucleotide sequence. Local alignment tools like the BLAST family detect these specific blocks of sequence similarity, ignoring the noise between them. Once a local similarity is found, global alignment tools like Procrustes or GeneWise are deployed to infer the complete structural boundaries of the gene. Similarity searches are highly accurate, but they inherently rely on history. They only work if evolution has already shown us what to look for in another organism. This creates an inherent limitation. When researchers sequence an entirely novel organism or discover a unique functional gene, historical comparisons break down. If a newly sequenced gene lacks any close relatives in existing biological databases, similarity alignment tools will return absolute silence. To discover entirely new biological functions, computational biologists cannot rely on comparative history. They must rely on raw mathematics. The failure of database matches necessitates an entirely different algorithmic approach, one capable of predicting a gene from scratch using only the sequence itself. This is ab initio prediction, a Latin term meaning from the beginning. Ab initio algorithms completely ignore external databases. Instead, they scan for intrinsic statistical properties hidden within the DNA itself. This prediction requires two critical data inputs. The first is signal sensors. This diagram outlines a coding region, identifying structural landmarks like the Tata box. Signal sensors hunt for these deterministic motifs, acting as physical boundaries. In eukaryotes, they detect complex signals, like splice sites at intron boundaries, defining where transcripts cut. Finding these signals is only half the battle. The algorithm also requires a second input, content sensors. While signal sensors provide the structural boundaries, content sensors must provide the statistical proof that the region between those boundaries actually encodes a protein. Content sensors evaluate the sequence by looking for patterns of codon usage bias that are entirely unique to a specific species. A codon is a sequence of 3 DNA or RNA nucleotides that corresponds with a specific amino acid. This chart compares the highly structured, biased frequency of specific codons in an exon against a flat, random distribution of sequences in an intergenic region. Because biological systems naturally prefer certain codons over others, valid coding sequences possess a distinct, measurable statistical signature. In eukaryotes, these distinct statistical signatures are heavily fragmented by random introns, making simple linear parsing impossible. Parsing this fragmented data requires advanced mathematical architectures, specifically hidden Markov models, or HMMs. An HMM is a statistical model used to calculate probabilities in randomly changing systems, where the internal states are hidden from the observer. HMMs continuously weigh the overlapping probabilities of all detected signal and content sensors simultaneously. This calculates the most mathematically viable path of a complete gene. Ab initio tools like GenScan calculate the highest probability of a gene's existence using raw sequence statistics alone. Relying purely on mathematical probability has a critical flaw. Ab initio methods suffer from high false positive rates due to the sheer vastness of eukaryotic genomes. There is a specific biological reality that frequently breaks these probabilistic models: alternative splicing. 40 to 60% of all eukaryotic genes do not code for just one single protein, but multiple distinct variants. This diagram illustrates alternative splicing branching a pre-RMNA transcript. The cell dynamically rearranges exons to create entirely different functional instructions, resulting in 3 distinct RMNA sequences from the same DNA. These overlapping logics confound algorithms. In translation, these arrangements produce structurally distinct final outputs. Predicting a standard open reading frame is vastly insufficient if the algorithm cannot predict how the cell will actively slice and reconnect that exact same frame in real time. Because neither method is perfect in isolation, the modern gold standard for gene prediction is an integrated, evidence-based pipeline. Modern bioinformatics pipelines first deploy ab initio models to generate strong statistical hypotheses about where a gene and its boundaries might exist. Researchers then validate these mathematical guesses using real-time transcriptomics, specifically RNA-seq tools. RNA-seq physically sequences the active RNA molecules present inside a cell. RNA-seq maps actual, physically expressed RNA fragments directly back onto the reference genome to verify activity. Mapping these expressed reads provides empirical proof of exact exon-intron boundaries and reliably detects complex alternative splicing variants that the math missed. To handle this massive data integration, bioinformaticians rely on tools like TopHat, HisSat2, or cufflinks. Robust gene prediction results from the synthesis of probabilistic modeling, evolutionary history, and empirical real-time expression data.